So, you really think I'm just over-interpreting and putting too much meaning into everything she says? Oh, man. Oh, hi. I didn't see you there. I'm Dr. Pettus. You caught me confiding in a friend. But I'm glad you stopped by, because I want to tell you about the lab you're going to be doing tomorrow and help get you started off in the right direction on that. So let's switch over to screen share. I'm going to go to my laptop right over here. Hold on just a second. Here we go. So, my fellow anatomists and neuroscientists, you have access to the PowerPoint presentation of these entire lectures. The purpose of this is to give you a compact, condensed picture of a few things that I think will help you in lab tomorrow. Not only that, but I'm going to try and get to some of the more difficult ones. I mean, I know some of this stuff you've had it before, so even if you don't remember, it's easier to relearn it if you've already known it before. So here we go. All right. This you've had before, but I want to do it again because it's so important. So of course we talk about dorsal being posterior, ventral being anterior. When we're talking about the central nervous system, we also talk about rostral and caudal. Rostral being toward the nose, caudal being toward the tail. So that's pretty easy to do in a rat. In the humanoids, it's a little more difficult because being bipedal, we kind of bent our heads here, and the central nervous system takes a bend also. So when we're up here in the cerebrum and the diencephalon, we'll know what that means in a moment. Rostral, once again, toward the nose, but caudal toward the back of the head. And once we're down here in the brainstem, we talk about rostral as being toward the cerebrum and caudal being toward the tail. And once you understand where the brain sort of takes a bend right here, and we'll be seeing that, it does make a little more sense. So really, caudal down toward the tail, rostral still going up toward the nose, but let me take a turn right here. See? Okay, not so bad. What else? The rest of this, okay, so we're gonna talk about a sagittal plane. So that's where you separate left and right. We'll be doing that in lab. Coronal plane, we're gonna be doing this with the brains. That's where you separate front from back. So you can see left and right halves. And then there's the horizontal plane, which is separating the top from the bottom, separating superior from inferior. And all of these have their pros and cons for, when you're looking at different things. All right, keep moving. Basic organization of the brain. We can divide it in lots of different ways, but one way we do it is dividing it into 5 parts. So let's start at the bottom, actually. The spinal cord evolves into the medulla oblongata as one ascends rostrally, and it's part of the brainstem. So the other 2 parts of the brainstem are the pons and the midbrain. So this is the ventral side, this is the dorsal side. So it's easy to tell the pons. It's this I don't know, basilar body that sticks off the ventral side. So it's easy to see where the medulla becomes the pons, and it's also easy to see where the midbrain becomes the pons, also where they transition there. So, and then you go from the pons to the midbrain. That's the brainstem. Then we get to the diencephalon, which is an inclusive embryological term, and it includes the thalamus, the hypothalamus, and not shown here, and we won't talk about it, the epithalamus. Moving from the diencephalon onto the cerebrum. So the cerebrum involves what you probably hear very often is the cerebral cortex here as well. Now the cerebellum is off the dorsal side of the brainstem, and we'll see exactly how that lines up in a moment, but you can see here off the pons, see that kind of flat section right there? That's one place where the cerebellum attaches to the brainstem. Oops. Same thing, but what we have here is a midline view of a sagittal section. So if we cut right down the middle and separated the left and the right halves of the brain, we would be looking here at the medial side of the right half of the brain. So we can see cerebral cortex here and the diencephalon. This would be the hypothalamus here and the thalamus here. then onto the brainstem, including the midbrain. We can see where this bulge of the pons, the basilar pons, comes off there. So that's the pons. And where it ends, we've got medulla. And then it's really hard to tell just with the naked eye where medulla becomes spinal cord, but we'll talk about that in a moment. All right, so there's our basic intro, the parts of the brain. You want to have that down. You should actually be able to draw this. It's kind of easier from the lateral side. I don't have anything to draw with right here right now, but you just draw like a, kind of like that, and then come back to meet. That's how you do your cerebrum. And then you just accentuate, just draw a straight tube and put a basilar pons off the end of it for that. I'm sure Stefan Tiggs has an example of how to draw it somewhere out there. Okay, we're going to run through the meninges. I know I don't have to talk about this for long because you've already had this before. So the brain has a lot of protection. We go through skin, scalp, skull, down to meninges. The thickest, toughest outer layer is the dura mater. What does dura mater mean? That's right, tough mother. Everyone say it together, dura mater, tough mother. So that's tough like a, I don't know, like the skin on a football or something. And we go deep to that, we find the arachnoid matters, thin and wispy, but it also, like the dura mater, is more like a bag around the brain. And when you get the pia mater, it's more like a surgical glove, around every curve and every contour of the brain. It's pressed up against it. Can't see the pia mater in this picture. We just have dura and arachnoid. The CSF is found deep to the arachnoid in the subarachnoid space. So here we see the protection going down to the brain. So here's the brain. Let's work our way the other direction. So you have the pia mater right against the brain, and then you have arachnoid mater. Between the pia and the arachnoid, you have a little bit of open space there, and that's where the CSF is found. Then you got the thick dura mater lying up against the skull. Now you have spaces on the superficial and deep sides of these. So between the skull and the dura is called the epidural space. Deep to the dura mater, between in the arachnoid, is the subdural space, and deep to the arachnoid in here is the subarachnoid space. Now the only one of these that should exist in a healthy person is the subarachnoid space, which is filled with the cerebrospinal fluid. The epidural and the subdural space are potential spaces that get filled up when fluid leaks, in particular when a blood vessel is ruptured. You can see the details of that in the movie of the lecture, the full lecture. So back to the dura mater, just deep to the skull. So you might recall that there are two layers, your periosteal and your meningeal. So periosteal, of course, being against the bone. So you can see they're running together normally. But at some point, the meningeal can reflect off of the periosteal and sort of meet itself and form a double layer feature inside the skull. We'll learn a few of these. The one shown here is the falx cerebri, which divides the left and right cerebral hemispheres. Now, between the meningeal and the periosteal layer is where you find the, in this case, the superior sagittal sinus, but there are other venous dural sinuses, or I should say dural venous sinuses, that carry venous blood back to directly or indirectly to the jugular vein, internal jugular vein. Here we can see that falx cerebri. So there's a sagittal section just past the midline. So you can see the falx cerebri separating the hemispheres. And you can also see that superior sagittal sinus, so a triangular thing against the skull there where venous blood is flowing. When we look down into the skull where the brain has been removed, we can see this double layer of dura mater, formed just like this. But in this case, you can see it's separating the middle from the posterior cranial fossa here. And so it would be separating the occipital lobe from the cerebellum down below. That's called the tentorium cerebelli, or the tent over the cerebellum. You can also see where these, where there's a double layer of dura mater where it meets the skull, there's always going to be a dural venous sinus right here. Here's one here, and there's one. All right. You know, I mentioned how the cerebrospinal fluid is deep to the arachnoid mater. That's not where it gets its start. It gets its start in the ventricles. So let's learn the ventricles, and then we'll see how the CSF is formed in the ventricles and flows out to the subarachnoid mater. Here we see a magically translucent brain as drawn by Netter. And so you see the cerebrum here, cerebral cortex, cerebellum, brainstem down here. So in the cerebral hemispheres, there's a left and right cerebral hemisphere. So in this case, the ventricle we find in the cerebrum has a left and a right. These are the lateral ventricles. And you can see there's sort of a horn going into each lobe of the cerebral cortex here. I should say of the cerebrum. So there's a left and a right lateral ventricles that are in the cerebrum. Then they flow through an interventricular foramen. There's a left and a right interventricular foramen that go to the midline third ventricle. The third ventricle lies between the left and right diencephala. So that's right, it separates the left and the right thalamus. So there's only one of these, it's in the midline. So the only one where you have a left and a right where it's double is the lateral ventricle. All of these other ones are singular. So we get to the third ventricle via the interventricular foramen, and it gets to a very narrow sort of chute that goes through the midbrain called the cerebral aqueduct. Then we get to the fourth ventricle, which goes across a few brain regions. So on the ventral floor, we have the pons and the medulla, and for the roof on the dorsal side, the cerebellum. Take note, as I went through these, I mentioned a brain region, or in this case for the fourth, a few brain regions for each ventricle. This is going to be one of the more handy things you learn from this if you're not a devout neuroscientist, because these ventricles are very helpful in locating things on radiographs. So you want to learn these ventricles well, how to recognize them, and how to recognize the brain regions around them to help you read radiographs. And here is the key. You should be able to write this on your own. If I pass you in the hall and I name a brain region, I want you to name that ventricle, or the other way around. If I name the ventricle, you got to just boom, spit out the brain region. How does this stuff flow? Well, we start up here in the lateral ventricles, and so okay, so everything red in this picture is a choroid plexus, and that's a vascular specialization that makes the cerebrospinal fluid. So we find it up here in the lateral ventricle, we find it in the third ventricle, and we can find choroid plexus down in the fourth ventricle. So any ventricular space that has ventricle in the name has choroid plexus in it. What that means is that the cerebral aqueduct is the only one of these that doesn't have it. And you can't have it there because it's too narrow, and it would totally block the flow. It would clog up the flow of the CSF if you try to fit a choroid plexus in there. So we're going to be making the CSF up here in the lateral ventricles, and these are cul-de-sacs. So if you're making it, there's only one way that pressure can escape, and that's going to be through the interventricular foramen into the third ventricle, where you're still making the stuff. So that's going to build pressure and push the flow through the cerebral aqueduct into the fourth ventricle, where you're making more of it, and it's going to push it then out into the subarachnoid space. Now it's labeled on the previous slide, but so you can go back there and look at it, but there's a median and two lateral foramen that allow for the escape of the CSF into the subarachnoid space. In the old days, they were the Luschka for lateral and Magendai for medial, but you know, we don't really say the people's names anymore. Let's see. There you go. I said foramen, sorry, it's aperture. Lateral and median apertures. So all right. So we escape into the subarachnoid space, and then so the CSF is floating all around the brains. So your brain is really, you know, floating in a vessel, just like in all those science fiction things where there's someone's brain preserved, floating in fluid there. And then you'll find, we're going to see this, along the dura mater, we'll see these white, crunchy little bumps on the most superior surface along the superior sagittal sinus. Those are called arachnoid villi, and it's where specialized, you know, arachnoid mater kind of bubbles through the dura mater into the superior sagittal sinus, and there are specializations that allow for the CSF to filter back into the venous blood of the superior sagittal sinus. So that's our arachnoid villi, or arachnoid granulations that do that. All right, and one last thing about CSF. I really show you this just because I want you to see what it looks like in a coronal section. That's how you're going to see this as clinicians. So if we look over here at the magically translucent brain, you can see how the lateral ventricle is C-shaped here. And so a lot of these coronal sections, you can have like a superior portion of it from the, here in this case, the parietal lobe, and down here in the temporal lobe. So if we look up here, this is lateral ventricle, and this is lateral ventricle down in the temporal lobe. So these are the same structure, you just happen to be sectioning it in an area where they have been separated, but you can see they're connected there. So this is the lateral ventricle in the parietal lobe right here, and we've got it right at the level of that interventricular foramen. And you can see right here, the third ventricle. Now, without even knowing any neuroscience, I saw a table earlier in this PowerPoint that told me the tissue around the third ventricle is going to be diencephalon. So this is going to be thalamus here. I know that because that's where it is. Okay. All right, we'll come back to cross sections in a little while. Let's look at the surface anatomy a little bit. Well, I'm going to start with a cross section. All right, ignore this for right now. Let's look at these at this section right here. So when we get up into the cerebrum, let's look over here first, excuse me. There is the part on the surface. These layers on the surface are called the cerebral cortex. Just like, you learned in the kidney, the cortex on the surface, the medulla down deep. So cerebral cortex out here on the surface. In the cerebrum, in the cerebral cortex, it's different than the rest of the nervous system in that you see the gray matter on the surface. So this is the gray matter, and that means you're going to have cell bodies there. And deep to that is the white matter where you have the axons. Well, also buried deep in the cerebrum here, you can see color-coded in pink here are some of the deep nuclei of the cerebrum. So gray matter on the surface is called cortex, and they're called nuclei deep in the cerebrum. That's your gray matter. Now, when we look at the cortex, you can see there are sorts of ridges and valleys. Your ridges are all called gyri or gyrus singular, and they're separated by valleys called sulci or sulcus singular. In some cases, there are super deep valleys that separate entire brain regions, and those are called fissures. So you might see, lots of sulci along here. Here's one that's named the central sulcus that actually separates brain regions also, but it's not that deep. And then you've got this lateral fissure separating the temporal lobe from other things up here. Examples to come. All right, this is a lateral view of the left half of the cerebrum. This is anterior, this is posterior. So here behind the frontal bone, you have the frontal lobe separated from what's behind the parietal bone, the parietal lobe. It's separated by the central sulcus, a sulcus you're going to want to identify in lab. The main way you can identify it from all these other sulci is you're going to find an entire gyrus from top to bottom down to the lateral fissure here. So I don't know, I learned this as fissure. Lateral fissure separates off the temporal lobe, and then separating the occipital lobe, you have a parieto-occipital sulcus. Great name, right? Between the parietal lobe and the occipital, but also that one. Now, this sulcus does not show up very clearly in the lateral view, but there is like a little dip you can see on the edge here and here, and then you can draw an arbitrary line. When we look at the medial side, you'll see this sulcus is as clear as day, but on the lateral side, not as clear. And then when we see the whole brain and the left and the right hemispheres, they are separated from each other, the left and right, by the longitudinal fissure. Now there's another lobe, kind of secret lobe. If you pull apart the frontal or parietal and the temporal lobe from one another, you'll see some gyri deep between them. That's the insular cortex. You can see it here. So here's your temporal lobe. This is like, this thing is pulling on that right there. We're pulling up on there. And so you'd see down in here another lobe here called the insular cortex. And then deep to that, you can see these nuclei, these deep nuclei of the cerebrum. All right, back to the surface anatomy here. Here's that central sulcus. And so in front of it, anterior, rostral, we find the precentral gyrus. Good name, right? In front of the central sulcus, precentral gyrus. Behind it, we see the postcentral gyrus. Now, knowing the function, so we can name it for its location, but we can also name it for its function. Anterior or rostral to that central sulcus, the precentral gyrus, is the primary motor cortex. So this is the last stop for motor messages going down to the spinal cord. The cell bodies that you find in the premotor cortex here, their axons travel all the way down to the spinal cord to the level of the muscle that's going to be innervated. So it goes to the nerve, like the next one down the spinal cord projects to the actual muscle. Posterior caudal to the central sulcus, we have the postcentral gyrus, and that is primary somatosensory. And when we say primary for sensory, it means it's the first stop in the cerebral cortex on the way in. So, right, so first stop, very fundamental processing there. Coming on back here, we have the occipital cortex we talked about earlier. It's going to be primary visual. The primary visual is really primarily on the medial side, but just the tip of it here is also primary visual. And then also over here along the temporal lobe, we have primary auditory cortex. So a couple things I want to point out about the somatosensory cortex here. So we just have this big gyrus with all this somatosensory information coming in from the outside world. And I want you to note that it is somatotopically organized in that certain regions of the body are represented in a map form across this gyrus, so that the feet are up top and along the medial aspect of the ridge there, then the body, then down to the hand and the face. Now I want you to take note that the majority of this surface area here is actually occupied by hand and face, because you know we have a great deal more detailed innervation of the hands and face. You have a much more sensitive somatosensory perception there. All right, the other thing is that what we see is contralateral representation, meaning the opposite side of the body, so that this left hemisphere of the cerebrum is representing the right side of the body. Two fundamental principles of the cerebrum there. So here's a medial view, and surrounded in red here, we see the primary visual cortex. It surrounds a sulcus called the calcarine sulcus. And your visual fields are somatotopically mapped also, such that this inferior aspect below the calcarine sulcus actually represents your superior visual field, and the opposite for this part above the calcarine sulcus. Motor, I could basically just say the same thing as for somatosensory, and that you have somatotopic mapping, so that foot muscles are innervated by cortex up here, face muscles by the more lower aspect down toward the longitudinal fissure. And it is also the same sort of thing that the hand and the face get a great deal more representation because you're a lot more coordinated with your hand. I've only seen like one or two artists ever play a piano with their butt, but they can, even those guys can play it much better with their hands. Also contralaterally represented, so that this left cerebral hemisphere, primary motor cortex, controls muscles on the right side of the body, contralateral. So use these words contralateral and ipsilateral as often as you can. You're going to be exposed to them, so you got to process them quickly. You ought to use them quickly to get you there faster. Okay, skipping ahead for what we're going to use in lab, let's look at the internal structures of the brain. So here's a refresher of what we saw earlier. You had the 5 divisions. So let's look over here where we have the magically translucent cerebral cortex. See, we don't really see the diencephalon unless we've cut the brain up. You can see maybe a little bit over here on an anterior view, but not much. So just think about it. When we're doing, say, coronal sections, you can do sections out here that'll be nothing but cerebrum. You see, you got to be sectioning all the way back here if you're going to include some of these brains, some of these deep cerebral structures, or I'm sorry, those aren't deep cerebral structures. That's diencephalon. If you're going to include the diencephalon, you got to be a little further back to where you see purple here. And then even a little further, you'll start to see brainstem. Another tricky thing to keep in mind, though, is that when you're doing coronal sections of the cerebrum, your sections of the brainstem are more or less longitudinal, right? So it'd be like cutting the brainstem this way. And a lot of us, who learn the brain, we get used to looking at brainstem sections that are sectioned this way. And honestly, I learned it. I learned brainstem sections this way, and I learned cerebral sections this way. So it's tough to put all that together. All right, so here is a coronal section of the cerebrum. And you can already see thalamus here in purple there. You can see you've got some diencephalon in this slice. So here outlined with the dotted line, you see the diencephalon. So I really just wanted to show you this, to show you this concept of how you end up sectioning the cerebrum and the diencephalon in one. We're going to look at some other cross sections for the details here. Okay, so that's for the diencephalon. Now there's some other important structures you're going to be spending a lot of time with over the next few weeks. And those are some of these deeper nuclei of the cerebrum. The ones you're going to hear so much about are what are collectively called the basal ganglia. And some of the things included in the basal ganglia are the putamen and the caudate nucleus. And I want you to see they're shaped kind of like the lateral ventricle. And as a matter of fact, these follow the lateral ventricle. So you're going to learn that it's very helpful for you to know that lateral ventricle to identify these nuclei that you're going to hear so much about. So putamen and caudate nucleus. Now there's another part of the basal ganglia called the globus pallidus. I guess in this picture, it's the green part there, but it's going to be in the putamen is larger than the globus pallidus. And so from this point of view, from lateral, you wouldn't be able to see it. It'd be obscured. But we're going to see lots of sections that include them, and you'll learn their relationship that way. All right, so we're going to be sectioning through these two lines right here and looking at the caudate, putamen, and globus pallidus and their relationship to the ventricles. So ventricles are easy. Let's start with those. We know along the midline in this region, we're going to see the third ventricle, because that's the first midline one that separates the left and right diencephalon. And then you have your lateral ventricles here. They're just under the corpus callosum you'll be learning about. And then over here in the temporal lobe, you also have a little bit of lateral ventricle. Now, here we have the diencephalon on either side of the third ventricle. Now, the way we find these basal ganglia elements is using a combination of the lateral ventricle and what's right here, the internal capsule. You'll learn the details on exactly what that is later, but for right now, just know that it's a mass of white matter just between the caudate putamen and the caudate and the putamen, between the caudate and the putamen. So we use these ventricles to find the caudate nucleus. It's always bumped right up against the lateral ventricle. And then as you go lateral to that, you'll see this white matter, and you'll see another nucleus lateral to that, the putamen. Then medial to that, the globus pallidus. Now, of course, these are colored pink, so they stand out, but you'll be amazed how clear they are actually on the brains too. The white matter and the gray matter are quite distinct from one another. Okay, this is in that last picture with the nice color coding, this is what we were looking at. Let's try it on this more realistic section here. Over here, you have that lateral ventricle that we're not really talking about so much right now. And then you have the lateral ventricle up here, and you can see over in the corner of that lateral ventricle, that caudate nucleus. Then you got this wide swath of white matter. If we go across that from the caudate, you bump into the putamen. You see a thin little white line and a lighter nucleus right there. That's the globus pallidus. So there you've got three of your major elements of the basal ganglia that you'll be learning about right there. If we look up here, so what we were just looking at was this section. If we go further anterior right here, we're going to be anterior to the diencephalon. So there's no thalamus in this one. So here I have cerebral cortex. We have these deeper nuclei here. Here's lateral ventricle. And we are actually, there's a little bit of temporal lobe, but it's the tip of the temporal lobe. No more ventricle down there. So right up against the lateral ventricle up here, you got the caudate nucleus once again. You got some white matter. And then over here, you got your putamen. And you see that thin little white line there. Here's this little triangular shaped globus pallidus next to that. Alrighty then. So you can rewind and review that. We're going to look at the same things now, though in a transected brain. All right. So here are our ventricles. We've got lateral ventricles. We know they're lateral ventricles because there's a left and a right, correct? And where they come together, and you just see one in the midline, well, you know that's going to be the third ventricle. And so you also know that on either side of the third ventricle, you see thalamus. Riding along lateral to the lateral ventricle is the caudate nucleus. And we can jump across the white matter to the more lateral putamen, and on its medial side, the globus pallidus. Ta-da! So you guys are experts on reading radiographs now. I just want to point out one other thing you can see in this section, and you can go back to the coronal sections and identify this as well. But if we go in here between the temporal lobe, if we go in the lateral fissure here, you'll see the insula. And so I want you to appreciate where the insula is relative to the putamen and the globus pallidus and the caudate. Now, a lot of people, there are scientists who study the basal ganglia, and they can identify these things on these sections. But holding a whole brain in your hand, it's very difficult to understand exactly where that is. But you guys will always know, because you're going to pull that, if you pull that temporal lobe apart from whatever you're looking at up here, the frontal or parietal lobe, you'll see and you'll bump up against this insular cortex. And you'll always know that if you just go like a centimeter deeper, you'll be starting with the basal ganglia there. So use that insula and this lateral fissure to identify whereabouts your basal ganglia nuclei would be. All right, I'm going to stop it at that. I will point out here, when you look in some atlases, they use a fabulous stain. It works well, but it can confuse beginners. It stains myelin. So what it means is that in these sections, the white matter is black or dark, and the gray matter is light, relatively speaking. So just be aware of that. And in a beautiful picture like this, you're looking at dark white matter. So here's this insular cortex. Here's that putamen just deep to it. You got the globus pallidus right there, just medial to that. And then you got some white matter that's stained darkly. Some more white matter here, stained darkly. And there's your caudate bumping right up against the lateral ventricle. And here's your thalamus bumping right up against the third. So here's the trick. You don't know what all these brain regions do. You're going to. But you want to get a feeling for identifying and finding them and toss the words around. So when you get to the neuroscientists and the neurologists who are telling you about this, they're going to be boom, 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 reeling off lentiform, basal ganglia, thalamus, putamen, globus pallidus, caudate, insula, much faster than I just did. And I want you to be familiar with it and have a visualization of where these are as you're adding meaning to them. All right, thank you, and I'll see you in lab tomorrow.